Hello ladies and gentlemen, and welcome to a revision video. If you remember back to May, June last year, you'll have realised that we brought out a video entitled Biology AS Unit 1 Whole Spec Revision. Basically that video was really long, and so what we're going to do is we're going to bring back these videos, only we're going to break them down into more specific topics. So we're going to start off here with Biology AS Unit 1 Molecules and Cells, specifically the water, inorganic ions and carbohydrates. So we're just going to break it down so it's a lot more easier and manageable. We're going to go through a PowerPoint that I made myself for my own revision notes, and we're just going to go fast forward through this. So we're going to start off with water. So as you can see, the elements present in the largest proportions in the living organisms are hydrogen, oxygen, nitrogen, phosphorus, sulfur and carbon. The highlighted bits are the really key parts that the C.A.R spec will test you on mostly in terms of more knowledge, but you do need to be careful with the questions they ask you. That's someone that I had a lot of bother with. So the atoms of elements combine forming the biologically important molecules, and these are linked by the strong covalent bonds. We know this. We know water is a dipolar molecule, but what does dipolar actually mean? Well, it means that oxygen is slightly negative, but the hydrogen is slightly positive. These hydrogen bonds are weak bonds. Water has a polar nature, and it results in the formation of these weak hydrogen bonds that are slightly positive between the water molecules. But the hydrogen bond is found between the oxygen and one water molecule and a hydrogen atom on the other. Water is a very good solvent. Salts and sugars and amino acids dissolve readily in water, and this is called hydrophilic. So hydrophilic means water-loving, whilst non-polar molecules such as lipids do not dissolve in water, and this is hydrophobic. Hydrophobic is water-hating. So overall, water is capable of dissolving a wide range of chemical substances, and ions are charged and are all surrounded by shells of water, known as water clusters, around which charged groups of glucose and amino acids. Biochemical reactions within the cell are carried out in a solution, and water is used to transport nutrients. The hydrogen bonding of the water molecules is known as cohesion. The energy required to break the hydrogen bonding in the liquid water is the latent heat of evaporation. Moving on to inorganic ions. So in this table you can see a list of all the ions and then an essential component of them. This could be later on important when trying to tie it into a question. So we're just going to go through them. Calcium has an essential component of middle lamella, and this is in the plant cell wall and bones and teeth in mammals. You'll know about the middle lamella of the plant cell wall later on down the line. Hemoglobin is important in iron. You'll also find that one later on down the line too. Magnesium is important in giving chlorophyll. Potassium is important in maintaining electrical gradients across neurons. Nitrate is important in free amino acids, nucleic acid and chlorophyll. Phosphate, that's good for phospholipids, and the phosphate main component of the cell membrane. Again, we'll focus on that later on. And hydrogen carbonate, this is important as a buffer. You may ask, what is a buffer? Well, there it is, just below the table. A buffer is a chemical or substance that resists a change to pH, such as hydrogen carbonate and albumin. An osmotic system as well, just for note, is the movement of water in relation to water potential, high to low concentration. We'll go on to osmosis later on as well. So carbohydrates. We'll start off by telling you what an organic molecule is, and this is a complex carbon-containing molecule formed of subunits called monomers. They join together forming large polymers through the process of polymerization. Carbohydrates contain carbon and hydrogen and oxygen only, but carbohydrates are split into three groups called monosaccharides, disaccharides and polysaccharides. To start off with, we're going to talk about monosaccharides. So the general formula is CH2O and then N, and this N varies from about 3 to 9, but you don't really need, they won't go in depth with this. So you go triosis, which is N3, pentosis N5, hexosis N6, etc., etc., etc. Monosaccharides are soluble in water, they're sweet and crystalline, and the most common monosaccharide is glucose, which is C6H12O6. There's two types of glucose, alpha glucose and beta glucose, and these are important, because alpha and beta glucose are isomers, as they have the same molecular formula but different structural formula. Alpha glucose is the basic subunit of starch and glycogen, whereas beta glucose is a constituent monomer of cellulose. Fructose, which is another one that you could be asked on, is a simple monosaccharide found in many plants, and fructose is derived from sugarcane, sugar beets and corn, just to name a few. Moving on to disaccharides. So their general formula is C12H22O11. You can see the double amount of hydrogens there as the oxygen and carbons. So they dissolve in water, producing a sweet tasting solution, and they're formed when two monosaccharides react together as a condensation reaction, which is the removing of water. A bond is formed in this reaction, and it's called a 1,4-glycosidic bond, and this is formed between carbon 1 and carbon 4. It's a reversible reaction, so disaccharide molecules can be broken down into monosaccharide monomers in a hydrolysis reaction, which adds water. There's a diagram of the molecular structural formula of maltose down there. If we go back as well, you'll also see the alpha and beta glucose. If you were to actually draw them, you'll be able to see where the circle is, about the difference, the key difference in it, and the picture below is a structural formula of fructose. So now moving on now to polysaccharides. This is only an introduction because polysaccharides is quite large. They are many monomers. They have a general formula of C6H10O5, and they're formed through a condensation reaction also. They're not sugar, so they're not sweet, and they are insoluble in water. But the important polysaccharides are starch, glycogen, and cellulose. And if you remember back, that glucose is very important in making these up between alpha and beta. So find out the difference now, because we'll move on to starch. So starch is a storage polymer of alpha glucose, and amylopectin and amylose are two types, with amylopectin being 80% of starch and amylose 20%. Amylose is alpha glucose molecules linked by a 1,4 glycosidic bond coiled forming a spiral, and these are held in place by hydrogen bonds. Whilst amylopectin is an alpha glucose molecule linked by 1,4 and 1,6 glycosidic bonds, with the 1,6 occurring every 20, 40, 30 glucose units. This is important because if you were asked to tell the difference, you would have to state that amylopectin has a 1,6 glycosidic bond, whereas amylose doesn't. The benefits of starch is that they're very compact and they contain a rich store of glucose in a small space. They are insoluble in water, so this does not affect the water relations of the cell, but they are also large molecules, so they don't easily pass through the cell membrane, which is also positive. Moving on to glycogen. These are also made up of alpha glucose with a 1,6 and a 1,4 glycosidic bond. There's no two types, there's just glycogen, and of course you have to have the 1,4 and 1,6 for this. The benefits of that there is that they're storage carbohydrates found in animals and fungus cells. They're stored in the liver and muscle cells in mammals. They're compact and insoluble, so they don't affect water relations, but they actually have more terminal ends in starch on the branches, so hydrolysis occurs faster. Polysaccharides in general, they're adapted well because many glucose molecules can be stored in the cell. They're readily hydrolyzed. They're branched with many terminals. They're insoluble, so they cannot move out of the cell, and they're also osmotically inert. Moving on to cellulose, which is the different one because it isn't actually a storage polysaccharide. Its role is actually structural, and it is made up of beta glucose, and it has a 1,4 glycosidic bond with the alternate molecules rotated. So if you look down at the picture below, you'll be able to see that on the bottom of the first molecule, you'll see CH2OH, and then the next one, that's actually flipped on the top, and that's so you can get the bonding between them of the O and the H's. So yeah, so it's made up of beta glucose and it has a 1,4 glycosidic bond. The rotation is needed due to a different structure of beta glucose. If you go all the way back, you'll know that it's flipped. So the adjacent cellulose molecules are linked by hydrogen bonds, and these are grouped producing microfibrils. And each microfibril consists of hundreds of cellulose molecules. So, and the structure gives an immense tensile strength. The mesh to form the cell wall prevents the cell from bursting, so you'll know that the cell wall is made from cellulose. Thank you for watching our PowerPoint on water, inorganic ions, and carbohydrates. We hope you. We hope this new format makes it easier for you to revise this. And I've been Dylan from C.A.R. Entertainment, and I hope you enjoyed this video, and we'll see you for the next one.